Okay, guys, so we're going to be looking at the stages of mitosis, the process of mitosis. Now, for your reference, your textbook reference is pages 360 to 361. Now, your textbook doesn't go in a great amount of detail into the process of mitosis, and in fact, it doesn't have like a series of detailed diagrams like what I'm about to show you here. It does have detailed diagrams of meiosis, though, in chapter 10, but for mitosis, there's not a lot. But you do need to know some particular features of the process of mitosis. You need to be able to describe like what is happening to certain structures during mitosis. So there are a few key structures that I want to draw your attention to. The first three are the chromosomes, the spindle fibres, and the centrosomes. You need to be able to make reference to these three different structures during mitosis. Now, at different stages, there are other structures that you can refer to. We'll get to those as we go through each of the stages, but you must be able to refer to the chromosomes, the spindle fibres, and the centrosomes, and we'll go over what those are at each step. So here are the goals. We're going to be able to hopefully name in order the phases, draw diagrams for each of these. Now these are for me drawn out already. I will be labeling them, but I want you to be able to draw them and label them yourself. Finally, you have to be able to describe in dot points the key features of each of the phases of mitosis, making reference to those key structures that I described previously. So before mitosis begins, we are in interphase. So think back to the cell cycle. We have interphase forming the main part of the life of the cell. And interphase prepares the cell for mitosis by we see the replication of DNA. We see the extra organelles being produced, the centrosomes being doubled. So I'm just going to label some of the things here. So we have here, these here are the centrosomes. Now the centrosomes are where the spindle fibres originate from. They're like your headquarters for the spindle fibres. Now the centrosomes, they're made up of two smaller subunits, right? These are two identical subunits of protein, and these are called the centrioles. So people say that centrosomes are made up of 2 times centrioles, and people often interchange the two terms. So centrosomes can be used or centrioles can be used. I prefer to use centrosomes, so that's what I'll be referring to them as going forward. So our centrosomes are where the spindle fibres originate from. And we do see some of the spindle fibres coming out of the centrosomes here. So you see like the spindle fibres, they're very short, but they're there. And they're coming out of those centrosome structures. We have labeled here this little green thing. This green thing here, I've now highlighted it in black, is the nucleolus. Now this is like a structure in the nucleus where the ribosomes begin their formation. The ribosomes are like the protein factories. Now we won't refer to the nucleolus beyond this diagram, but it's good to know what that is at the very least. Now what do we see here? We see here this green ring here is the nuclear membrane. And the nuclear membrane is completely intact at the end of mitosis. We also have here these stringy things. These are our chromosomes. And if you want to add more detail, these chromosomes are replicated chromosomes. So each chromosome has 2 chromatids attached firmly together. Hence why we call them replicated chromosomes. Okay. And the reason why they're replicated is soon we're going to be separating them apart into and distributing them into 2 cells. All right, so this is pre-mitosis, in the late G2 stage. Now we move into mitosis, and the first phase of mitosis, and I'll just write the acronym for mitosis, PMAT. The first stage, the P, is prophase. Now, prophase, the beginning, we have to see certain things happening. First, we see, what do we see here? We see like the spindle fibers in interphase, you saw they're very short, right? Now we start to see that they're growing a little. So the spindle fibers, the spindle fibers are growing. The spindle fibers are growing. And while they're growing, we actually see that the centrosomes, these centrosomes here, are moving to opposite poles of the cell. So they're creeping along like the plasma membrane of the cell, moving to opposite ends of the cell, okay? I should note that on the left we have early prophase. This is early prophase on the left, and this is late prophase on the right. So you can see that in early prophase, those centrosomes are close together, like they were in interphase. In interphase. So if you look at interphase, you see that they're really close together. In early interphase, early prophase, they're starting to move further away from each other, and by late prophase, they're at opposite ends of the cell here. We also see that those spindle fibers at late prophase are like fully developed. Now, late prophase, some people say, is more like early metaphase, but there are some things that like happen during prophase that you can see by comparing the two diagrams, the early and late prophase diagrams. So you see that the nuclear membrane is fully intact at the beginning of prophase. Like at late interphase, we see that it's fully intact, right? At late prophase, you can see that the nuclear membrane is actually breaking down. So this is a key feature of prophase. You must be able to make reference to this fact that the nuclear membrane is breaking down. So during prophase, the nuclear membrane breaks down, dissolves. Okay, so the spindle fibers grow during prophase. The centrosomes move to opposite ends. What about the chromosomes? What happens with the chromosomes? Well, we can see that the chromosomes in interphase, they were like long, stringy noodles, yeah? And during prophase, we start to see condensation, condensing of chromosomes. And I think this arrow here is meant to show us that the nucleolus is gone. So I'm just going to add this in. During prophase, the nucleolus disappears, but those chromosomes are condensing. So if you have a look at early prophase and look at late prophase, we can see that those chromosomes have become denser and denser. So you can see fully condensed chromosomes. Fully condensed chromosomes. Okay? All right, so I've written at the bottom that you need to be able to describe what is happening with the key structures, particularly the chromosomes, or you could say chromatids. Now, these words don't mean the same thing, but you sometimes can refer to chromosomes as two chromatids and so on. Nuclear membranes, if it's relevant, the centromeres, spindle fibers, and the centrosomes. Now, centromeres is I would say only really relevant to describe during metaphase and anaphase. So we're going to go on to the next phase. So PMAT, second letter is M for metaphase. What happens during metaphase? Let's just label some key things here. First, we have our centrosomes. They're still on opposite poles. But what is really, really, really key are these chromosomes and how they're lined up. So we see here that the chromosomes are aligned at the center of the cell. Some people call this, this line in the center of the cell the equator. Some people call it the metaphase plate. There are a few names for this, but the chromosomes, we find them lined up at the center of the cell. And they're lined up in a way that the chromatids, so if you look at each chromosome being made up of 2 chromatids, so let's just look at this one here. This here is one chromatid facing one end of the cell, and it has a second chromatid here, which is going to go to the other end of the cell. All right. So they're lined up this way to ensure the even distribution of chromatids. Each cell must end up getting one chromatid from each chromosome. And we can see here the spindle fibres. Spindle fibres are you see here that the spindle fibres are fully extended. And I might want to add here that the spindle fibres, some of them are attached to the centromeres of the chromosomes. So some spindle fibres are attached to the centromeres of chromosomes. So we can't really see the centromeres here. Did I include a picture of the spindle fiber anatomy? No, I did not. I had another diagram which shows like chromosome up close with the centromeres and those spindle fibers attaching to those centromeres here. Now there are a few different kinds of spindle fibers. I might just make quick reference to them in this diagram. There are 3 kinds actually that we can talk about here. The ones that directly attach to chromosomes, we call them kinetochores. Kinetochores. They have a special name. So they attach directly to the spindle fibers, and they're the ones that, the centromeres, and they're the ones that pull the chromatids towards the centrosomes in Anaphase. We have here these other spindle fibers that like attach to the spindle fibers from the other centrosome. So if you look at this spindle fiber here, it originates from the top centrosome, and then it sort of like doesn't attach to a centromere, but it attaches to the spindle fiber that originates from the other centrosome. We call these the polar spindle fibers. They're polar because they're at the ends, and they like attach to the ends of the other spindle fiber from the opposite end, or they start from the poles, whichever way you like to think of it. And they're used to like anchor the spindle network together. So these are the polar spindle fibers. And there's one more kind. These are the least prominent of the spindle fibers. They're the smallest. We call these the astral spindle fibers. Because they make the centrosomes look like stars, with like all these things coming out. So astral bodies are like things in the night sky, and they make the centrosomes look very starry, because they just come out like in very small pieces out of the centrosome, and their role is to attach the centrosomes to the membrane. So the membrane is like the surface that the centrosome can attach itself to and stay in place. And we have those there. So we don't often make reference to those astral spindle fibers, nor the polar spindle fibers, but we often talk about the spindle fibers in terms of their role in connecting with the centromeres and the chromosomes, and then pulling them apart later. Okay, So we've talked about centrosomes, the spindle fibres, and the chromosomes. Those are the key structures. And we also made some extra references to things like the metaphase plate or the equator of the cell, and unpack the spindle fibres a little bit more. Okay, during anaphase, let's go through the key structures again. We have here the centrosomes. They're still fixed in place. We have the spindle fibres now, and we'll just talk about the kinetochore spindle fibres, the ones that attach to the centromeres. Spindle fibres are retracting here, retracting towards the centrosomes. And in doing so, they pull those chromatids, or chromosomes now, towards the poles of the cell. So you can say here that the chromosomes are being pulled to the poles of the cell. So this arrow here, I do believe, is pointing towards the polar spindle fibers. You can see that these polar spindle fibers are anchoring onto each other, and they're assisting in the movement of, or the movement of those chromosomes. I think they're growing, and in them growing, by their growing, these spindle fibers are increasing in length, which pushes those poles apart. So these polar spindle fibers are pushing the poles apart. So this is just a minor detail. The main thing is that we see that chromosomes are being pulled apart by those spindle fibers, those kinetochore spindle fibers retracting. Okay. All right, so that's anaphase. Let's look at telophase. So in PMAT, the last letter, T, telophase. Telophase, I like to think of it as like the reverse of prophase. So everything that you see happening in prophase happens in reverse during telophase. So like in prophase, we observe the breakdown of the nuclear membrane. During telophase, we see the reformation of the nuclear membrane. So nuclear membrane reforms. We see the spindle fibers starting to grow during prophase, but during telophase, we see the spindle fibers disappearing. Disappear. And we see the decondensing of the chromosomes. So chromosomes decondense. Okay, so we've talked about those three key structures. The spindle fibers, oh no, we haven't talked about centrosomes. Centrosomes are one of the key structures, but those centrosomes, they're not really, I think there's not much to say about the centrosomes really in telophase. They remain at the opposite poles of what is still one single cell, soon to be two cells. But I think the one structure I did want to talk about was, not a structure, but a feature of telophase was this section here. So we can see here the pinching in of the two cells to form what we call the cleavage furrow. So cleavage furrow is forming, and from this cleavage furrow we'll see like the plasma membranes coming together, and in cytokinesis you'll see that this is where the two cells will separate. Okay, well, we have a name for that cleavage furrow. So for animal cells at least, this is the cleavage furrow. Plant cells, they divide a bit differently. They form a cell wall plate instead. But for animal cells, we have this. All right. So remember, guys, you have to be able to describe the three key structures in every step. Chromosomes, centrosomes, or centrioles, and the spindle fibers. And for some of the steps, you'd have to refer to other details. Like here we have the cleavage furrow in telophase. In prophase, we have the, no, not prophase, but in metaphase, we have the metaphase plate. There was something else. Oh, you can talk about the nuclear membrane breaking down during prophase and reforming during telophase. And yeah, so I would now just like if you haven't yet already, just put a brief description, like 2 or 3 dot points for each of these phases here, just to put like put some words to this, not just labels to this diagram, but have the steps, and making reference to those key structures in each of the steps.